Hello, welcome to Converging Dialogues. This is Xavier Bonilla. On this episode, it is a big honor, big privilege to have on Dalton Conley. Dalton is a sociologist and professor of sociology at Princeton University. He's also a research associate at the National Bureau of Economic Research and faculty affiliate at the New York Genome Center. He has his bachelor's from the University of California, Berkeley, MPA in public policy, and PhD in sociology from Columbia University, as well as a second master's and second PhD, both in biology from New York University. Much of his research has focused on the intergenerational transmission of socioeconomic and health status from parents to children, impact of parental wealth and explaining racial attainment gaps, causal impact of birthweight on later health and educational outcomes, sibling differences appear to reflect the triumph of achievement over ascription, and finally, genetics as a driver of both social mobility and reproduction. He's won many awards, many fellowships, and he is the author of numerous books, including the most recent, The Social Genome, The New Science of Nature and Nurture. And we get all into it. We start by talking about this nature-nurture debate, what that is. Many people talk about it, nature, nurture, 50-50 kinds of things, kind of colloquially. So we talk about some of the truth and science about that. We talk about polygenic index scores, PGI. We talk about the ethics of PGI and the GWAS studies, the Genome-Wide Association studies. We also give some critiques of the bell curve. We talk about the three major gene-environmental interactions, active, passive, reactive, epigenetics, the social genome, and many other topics. I really, really enjoyed this conversation with Dalton. He's obviously very, very, very brilliant. He has done so much research in this area. And I think the one thing I really enjoyed about him and his research is, he's, sort of a straight shooter. He does not get into any of the sort of politics or any of the messy debates around these things in an inflammatory way. He sticks on the data, is not trying to, be too aggressive with things or with other people that may disagree. Really just sticks on the data and why it's important. And really his emphasis is on the sociality of things and how we as individuals impact other individuals in our society. So it's wonderful work, and the book is great. Of course, you can find this conversation and all other conversations at convergingdialogues.substack.com. I'm also on YouTube, so please subscribe over there. Appreciate everyone that does. Share widely with your friends. There are many other conversations on this topic and get into all the details. And so now I bring you Dalton Conley. I am here with Dalton Conley. Dalton, thank you so much for coming on the podcast. I'm excited to talk to you.
Me too. Thanks for having me, Xavier.
Yeah, for sure. You got a great book that is just out. It is The Social Genome, The New Science of Nature and Nurture. I've really enjoyed this. Of course, many people have written about these topics for many decades, and I really, really enjoyed how fresh it is on the science. I loved how it's a topic that people talk about often, and how it's really, really coming at it in a nice angle, in good earnest, good science that you've done here. So it's well researched. Really, really enjoyed it, and I think it will be a nice addition to conversation people are familiar with. So we'll get all into it. Before we do, just tell listeners who you are professionally, academically, all the relevant details for you.
Sure. My name's Dalton Conley, as you mentioned, and my day job is to be a professor of sociology and population studies at Princeton University. I'm also a research affiliate, faculty affiliate at the New York Genome Center, and that tells it all. I work at the intersection of social science and genetics to understand nature, nurture, and the roots of human behavior.
Well, I mean, it's certainly important. This isn't your first book. You've actually written a previous book on genomes and social genomics and things like that. And so, I guess this is your sixth book.
I think a couple more than that, but this is the second one on this little field that's emerging called sociogenomics. The
Uh-huh.
first one was published in 2017,
Uh-huh.
and it was with Princeton University Press. It was more academically oriented, and also this field is moving so fast. I mean, literally, I would say it's existed in one form or another for about 10 or 12 years. So
Yeah.
that book was written like half the lifetime ago of this field. So much has happened since then.
It's completely outdated at this point. It's only 5 years old or so, right, or 7 years old, whatever it is. Yeah, no, that's good though. That's good that you stay on the curve of things. So let's just dive right in. I think a lot of people, and for those that don't, well, they'll get this today. Most things for humans are polygenic, meaning that even things like fingernails and hair and height and all that have multiple genes involved. And so it's not this, I think after we did the human genome or whatever in the 90s, one gene for one trait or things like that. And it turns out that's not quite true. In fact, the opposite. Um, what is, I guess, when we're talking about, it's always been seen in terms of how people discuss or debate this as this kind of like purely genetic and, environmental, social, kind of cultural things. But if you have a bunch of genes involved in a variety of traits, isn't it all just going to be a mixture of genes and environment? Or can we know that some things are just kind of purely, quote unquote, genetic? What's the kind of right lens to look at that famous dichotomy of, nature nurture?
Yeah, that's a really good question, Xavier. But it's really the wrong question. As I argue in the book, which we've been fighting this nature nurture debate, which is more important, genes or the environments we happen to encounter. We've been fighting for about 150 years, a little more, since Francis Galton actually coined that term, nature versus nurture. But it turns out that there is a porous boundary. So,
Mm,
for example, first of all, how our genes matter uh, depends on the environmental context we're in. That's kind of been an old insight, that what we call gene by environment interaction. So, to give a concrete example, in the late 40s and about 1950, about half of Americans smoked cigarettes. And when we look at those cohorts of smokers, genes didn't tell us much about who smoked and who didn't smoke. It was kind of a social
mm,
phenomenon. We didn't know as much as we do now about the dangers of smoking. And then, boom, we get the 1964 Surgeon General's report that smoking causes cancer and can kill you. And you get more and more evidence and more interventions to make smoking more difficult, like cigarette taxes and bans in
mm,
bars and restaurants and so forth. Since then, and today, about 10% of Americans smoke, but who smokes and who doesn't smoke is much more predicted by your genes. So the genes simply just didn't matter as much back then, and now they matter a lot. They
mm,
matter so much so that uh, the cigarette taxes, as they were being raised over the course of decades by different states, they kind of shrank the pool of smokers. Some people are like, oh my God, I can't believe how expensive a pack of cigarettes are now. I'm going to quit, and they stayed quit. But today, when we raise cigarette taxes, it doesn't actually affect how many people smoke, because the residual pool of smokers is so hardwired genetically to smoke that you could raise that pack of cigarettes to $100 a pack, and they're going to still shell out that money. They have a very um, low price elasticity, as in the economic terms. So, how the genes for nicotine addiction matter completely or don't matter completely depends on the environment. That's number one. That how your genes matter depends on even like this historical context. But beyond that we've known for a while. But beyond that, now we're seeing that how your genes have their effects doesn't stop at the boundary of your skin. Like, it's really important um, for your genes to interact with the environment to create an environmental niche, if you will, to have their effect. So, to give you like an example that we tend to think of genes as like a blueprint for a building construction being constructed. That it's like the specs the architect drew out, and then it's going to be 20 stories. It's going to have 2 elevators. It's going to have this many units and so forth. And for like an outcome like height, which is highly polygenic, and we can get back to the question of polygenicity later. But that's a pretty good metaphor. Yeah, you might, have had a childhood illness, or you might have some environmental deprivation where you don't get enough nutrition, and it's going to shrink the size of the building, and that's going to be an environmental effect. But for the most part, your genes have laid out the plan, and they don't need much about the environment to make. They don't need to interact with the environment to realize the plan. But then take something like intelligence. Intelligence has the same adult heritability, meaning the same proportion of the variation in the population is due to genes as it is for height, more or less, a little bit less. But how it operates is very different, because in childhood the genetic influence is very low, but when you get to a fully grown adult, a mature adult, then the genetic influence has expressed itself. So it's not like there's a building architectural sketch that is just followed. What happens is more akin to machine learning or AI. You get this. Genetics gives you the tools, but what they say is go out and collect training data, and how you interact with the environment is the primary mechanism. As important as what kind of myelin sheaths you have on your neurons, or how good your sort of blood oxygenation in your brain is, all these like internal factors. The external factors are an as important part of the causal pathway to get from DNA to doing really well on the SATs or one of the IQ tests that you give. And so that training it just gives you an algorithm and says a greedy algorithm, go out and collect data, refine your skills, and that's why after a lifetime of data collection, we see the genetic more interaction with the environment, ironically, yields higher genetic influence on the trait, because the environment is so key to the causal pathways for the genes. So, to give a more concrete example of that, in our own research, we found that if you take 2 parents and let's say they're each at the 50th percentile for the genetics of educational success, and we can talk about that also, what that means exactly. But they could have a kid at the 40th percentile just by random chance of which genes they inherit from each parent, or they could have a kid at the 60th percentile. If they have 100 kids, they're going to average at the 50th percentile, like the average of the parents. But each kid is a different draw. They get the kid at the 60th percentile or even higher compared to the kid at the 40th percentile, they end up parenting that kid differently, because that kid's genes drive the social interaction with the parents differently. From as early as 18 months, that kid is demanding more, one more story at bedtime, one more book. The parents end up sort of engaging in verbal play with that kid more. So the kid is directing the kind of nurture that they get, unconsciously, of course, from their genes. There's been no report cards yet. There's been no, probably developmental exams by a pediatrician to tell the parents this kid is a genius. It's just this organic interaction driven by the genes of the kid that extract the kind of nurture that those genes need to come to their ultimate phenotype of like high achievement in school. So, but that interaction with the parents is a critical causal pathway. So you can't really separate out the genes from the environmental niche making that they're creating. So that's sort of that's step 2. Step 1 was the genes. The effective genes totally depend on the environment, and effective environment depends on genes. Step 2 is that the genes require the environmental pathways to realize their effect. And then the third part about how there's really no true boundary between nature and nurture is the fact that what we call the social environment is largely genetics 1 degree removed. In
Mm,
other words, the important people in your life that create the environment in which you're socialized, your parents, your sibling, your peers at school, um, they're in turn driven by their own genes partly, so that um, what their influences on you can be described as is called genetic nurture, to use one term, or social genetic effects is another term for it. So, one example of that is that in our research we found that um, in a given high school, let's say this year's 10th grade class doesn't have a lot of people who are predisposed genetically to smoke. But the next year's 10th grade class randomly got a handful of kids that are at the top end of the charts in terms of the genetic propensity to be addicted to nicotine. That second class is going to have much higher smoking rates. That those few kids that are hardwired to be addicted to nicotine are going to kind of start a genetically sparked wildfire that's going to spread through the social network. And even if I don't even personally know those kids, my likelihood of smoking is being driven higher by their genes.
mm,
The genes of those few kids are in fact so powerful that they're almost as powerful as the effect of my own genes into my body. So um, here we're talking about the social environment, but it's also the genes of others. So those are three ways in which we really can't even separate out nature and nurture, I would argue.
mm. Yeah, you outline it very nicely. I'm thinking about, and you kind of mentioned it in the book a bit about things like Huntington's disease, PKU, sickle cell. These are things that people typically will say this is just a kind of quote unquote pure, um, genetic deficit or deficiency that we can see that even if no matter what environment you're in, not to say that's not important, but if you have the Huntington gene, you got a 50, flip a coin, you have a 50% chance of having Huntington's. What do we make of things like that, where you see these deficits or these challenges with certain types of neurodegenerative disorders or other types of chromosomal abnormalities, things like that? What do we make about, again, not to say that environment isn't impacted, but that maybe the more front loaded on the genetic kind of reading of these things? Is that again not completely accurate? How do we make sense of that?
But there's definitely certain traits for which um, the environment doesn't matter much. Like
Mm,
let's take eye color.
mm,
It's controlled by a handful of genes, not I think two, if I remember correctly.
mm,
Um, and whether I grew up in um, Asia, North America, whether I grew up in great poverty or wealth, my eye color is going to turn out the same. It doesn't require. It's more like that building specs analogy I gave. That it doesn't require interaction with the
mm,
environment. It's just a trait that is almost wholly internally developed. There's some very extreme environmental conditions uh, that can actually affect your eye color.
mm,
Um, Kaiser Fleischer rings that because you're not you have a overload of copper in your bloodstream will actually change your eye color, for example.
mm,
But that's extreme. And you mentioned a couple other examples
mhm,
like ALS or Lou Gehrig's disease. Um, eh, Currently, there's no environmental intervention we know about that will affect the course of that disease. And most of the things we do know that affect whether you degenerate fast or slowly or age at onset um, are about other genes inside your body, and we don't know much about how the environment may affect the progress of that disease. But those are more the exception than the rule. They're the exception in many ways. They're very um, monogenic.
mm,
They're affected by one gene,
mhm,
and also um, they yeah they, that they're pretty environmentally resistant.
mm,
And most other conditions that we talk about and that are like determining population health and other population behaviors and so forth are, are highly polygenic. They're influenced by thousands of little genetic switches all across
mhm,
the 46 chromosomes. Um, everything from height we talked about already, but arthritis,
Hmm.
um, coronary artery disease, schizophrenia, and as recently as 2003, you mentioned the Human Genome Project. As recently as 2003, when that was completed, people thought, no less than James Watson, the co-discoverer of DNA, thought that pretty soon we would find in this quote unquote dictionary of life the handful of genes behind schizophrenia, the maybe 10 genes behind heart disease, and then pharmaceutical companies would get to work quickly and either mimic those effects of those genes if they were kind of turned off, or block them if they were over expressed, and we could solve these diseases. And it was with great despair that we learned that it wasn't a few genes or even 20. It was literally thousands,
Thousands.
entire genome
Hmm.
that controlled almost all these traits. All these genes are controlling so many different traits. There's not like, oh, these are the 5 genes that are related to circulation, and they're different than the 5 genes related to your brain.
Mm.
Another interesting factoid about the Human Genome Project was that there was a betting pool in the 90s when they were sequencing the first genome, but how many genes would humans have? And they already knew that, for example, lowly corn had 100,000 genes. So it turned out that nobody came even close to the correct answer, which is about 20,000.
Mm.
Everyone way overestimated. So I don't know if they gave out the money for the pool or not, but well, what that means is that all those 20,000 are doing so many jobs in our bodies that they're implicated in your brain, they're implicated in your liver, they're implicated in your bones
Mhm.
development, and so forth. So um, when we talk about polygenetic influence, we're really talking about almost omnigenic. There's one theory
Mm.
called omnigenic influence, um, that almost every gene is involved in almost everything. And when we develop what I'll call a polygenic index, which is the key tool of this new field, to predict an outcome, predict your height, or predict how far you're going to go in school, it's really just developing different weights for all your genes
Mm.
for a different outcome.
Mm. Yeah, I wanted to ask you about that. So it's talked about a lot. I've talked about it on the podcast before as well, is this polygenic index, your PGI. And some people have speculated that in the future, maybe in 5, 10, 15, 20 years, you're going to go get a blood test, and you're going to go check your cholesterol, and you're going to check your blood pressure, and you're going to check your liver enzymes, you're going to check your calcium levels, and you're going to get your PGI on there too. And like that's the future of where this can go, that everyone's going to get a PGI for certain things. Just tell us what that is, where it's at now, either scientifically or in research, and maybe what it is more client facing in medical practice, if at all. And what is the ceiling, as best you can say now in 2025, of PGI? Is it a high ceiling or not? I'm not asking you to be Nostradamus here, but just as you're kind of seeing this, like where is it going and where could it go? Whether that's good or bad, that's a different question. But just those kinds of things.
Yeah. So going back to that early optimism after the human genome was sequenced, that quickly turned to despair when we realized that we weren't going to have like a pill to make us all cognitively enhanced, like the Bradley Cooper movie from I think 2012,
Right, right.
Limitless, that was called,
Hmm. Hmm.
because there were just too many genes involved in all these processes to have a quick pharmaceutical fix.
Mm.
Um, um, Wegovy aside, and the BM, diabetes drugs that seem to be working miracles.
Hmm.
That's the exception to the rule.
Right.
But the consolation prize was that in 2009, we got the first polygenic score, polygenic index, genome-wide risk score. There's a lot of different names for it. I use
Right.
polygenic index.
Hmm.
Um, uh, but where yes, there's like thousands of genes implicated across the entire genome, and they're all little effects. But what happens if we just sum all those effects? Um, it turns out, yeah, we can't like gene edit or use pharmaceuticals for those thousand of effects, but we can do a pretty good job of prediction. And so um, today, for example, I could take a swab of your cheek or a saliva sample or a blood spot, and I could predict your height plus or minus an inch, let's say.
Mm.
If I wanted to predict your education level, like if you're in the bottom 10 percentile of the education polygenic index, then you have a 7% chance that you graduated with a 4-year college degree. But if you're in the top 10%, you have a 71% chance.
Mhm.
So um, that sounds pretty stark and striking, but it's also very noisy. So
Mm.
there's these scores are still in their infancy. As we get more and more data, they improve, but there are some limits. So number one, how genetic a trait is going to be the ultimate upper boundary of how well something's going to predict. So
Mm.
height is thought to be kind of a good case study because it's really 80 to 85% driven by your genes in modern society where we're not having major famines
Mhm.
and other environmental um, forces that stunt us um, or such. And so right now, I think that the best polygenic index for height can predict about 50% of the variation.
Mm.
So we had more than half of what we could towards the ceiling, but we're not quite there yet. I've seen some papers that suggest that height, if we add more and more rare variation in the genome,
Mm.
then we get very close to we're getting closer and closer to the full genetic potential that you can predict.
Mm.
But for other traits, we're still pretty far from that. One I'm most familiar with or intimately is educational attainment. So we know from twin studies and adoption studies that how far you go in school is about 40% in your genome. Um, and Currently, the best polygenic index for education predicts about 17% of the variation.
Mm.
And even that is kind of an overestimate because of complicated reasons called um, population stratification. We could talk about later if you want. But the point is that upper limit will be about 40%. It's commonly thought that common variants that we actually measure when we do these studies, um, that means like that they're not rare new mutations
Mhm.
that are only in a few people. They probably cover about 25% of the variation in schooling. So we go from 40% to 25%, and that 25% is probably the upper bound for now for what we're going to hit. Um, but we're pretty far from that. But I think with more and more data and better techniques, we're going to approach that.
Mm.
What do you do with 25%? Um, it's it's obviously not deterministic, and that's part of the argument of the book is that um, you know how much you can predict and how you predict depends on the social environment.
Mm.
Um, uh, but 25% is still useful. Like if we're at 25% for cholesterol or for dyslexia, I think that could be very useful
Mm.
clinically.
Mm. Mm. Yeah, that's good.
Um, what I fear is that you asked about the social, how this is migrating out of the lab
Yeah.
and into the world. There's already in 2020 with the first PGI optimized baby was born, Aurea,
Mm.
and her parents. They don't say what PGIs they maximized, whether it was
Mm.
cognitive ability or minimized, depression risk, genetic risk, or what have you. But I would say that that's really getting over getting ahead of your skis, so to speak, because none of the scores predict so well that I would risk
Mm.
getting a sample from embryos. This was done in IVF, where they had a bunch of embryos, and then they sampled DNA from each of the embryos, and then calculated polygenic scores and decided which one to implant.
Mm.
And A number of IVF clinics are now offering this service routinely. So, if anyone's seen the 1997 sci-fi film with Ethan Hawke and Uma Thurman,
Gattaca.
Gattaca,
Yeah,
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 I mean,
yeah.
this is literally
Yeah, this is a
what
little plot line. Yeah.
um, so that's one application where it's already out of the lab and into clinical or social applications. The in Some cardiologists are screening young adults for their um, cardiovascular disease polygenic index very early, and if they are at the very high end, might start them on statins or other preventative measures from a young age before plaque starts accumulating in their arteries. That's another seems like a um, that seems compared to like quote unquote playing God with embryos. That seems like a more of a no brainer in terms of reducing cardiovascular disease. Then you go from there to, well, should long-term care insurance or even your car insurance company ask for your risk aversion um, uh, PGI or your dementia PGI, and should they adjust the rates based on that? Um, I think those are complicated debates, and they're ones we need to have sooner rather than later because there's been a lot of talk about germline gene editing, CRISPR. Are we going to have genetically modified humans? I think we could talk about it, but that's notwithstanding the two babies that were born in China a few years ago. That's not. We're not going to be swamped. Our preschools are not going to be swamped with genetically modified babies anytime soon for not just ethical reasons, but for technical reasons. But we do have this age of genetic prediction that has dawned, and we need to have much more discussion about what we want to do with that technology than we have had. We've had much more about CRISPR, which I think is more in the realm of sci-fi.
Yeah, it is. It is interesting because it's one of those things where, um, I think that, and I've heard some people make this argument where, I don't. It's an interesting argument that we have in our history of modern science in the 20th century, eugenics, which is a dirty word. And it was terrible things that were done there. It was, I think, between 1875, 1880 to like 1925, 1930. It was pretty standard. It was not of the day. It wasn't seen as a taboo thing. Now we've recognized it's like, hey, that was not good. We should not go back to that, and phrenology and things like that. But in some ways, people have suggested that IVF is a type of eugenics. I mean, you are selecting in and selecting out pre-labor and the whole thing as best you can. I mean, it does start to meddle a bit, and it's like, where is that line where, okay, you just gotta let it run its course. You can't factor out all the things you don't like, but then, or factor in, and it starts to get gray. It starts to get a little gray. People have made and some issues with stem cells, whether it's embryonic stem cells or whatever. There's interesting things that we're doing. Obviously, as you're talking about, CRISPR gene editing is another piece of it, and it's. I agree with you. I think we should have the debates. I don't think everything is eugenics. People just slap that on there and say, oh, says we don't talk about it, we don't do it. But it is interesting. I mean, what are the ethical implications of this? I don't know. I think that's why you got to talk about it. That's why you got to debate it. But it is interesting. Sort of connected to this, I want to ask about the GWAS studies. This will kind of come in there too. Is people have done these genome-wide association studies, and some people have. This is kind of back in the research area, so you can follow up to the ethics of modern day eugenics if you want. But the GWAS stuff is interesting because I've heard some people say, well, it doesn't really tell you anything, or it doesn't tell you as much as we want. And some people say, well, we'll just keep doing it and we'll figure out what it's going to tell us. And some people say, why waste your time? We're only getting certain kind of grant money and things like that. Like, it's not, I guess what do you feel are some of the challenges of leaning too heavy on GWAS studies? What are their limitations? But also, what are some of the things that they're good on? So you can answer my ethical modern day eugenics question if you want, or respond to it. But tell us about
I'll
GWAS
do
too.
both. So starting with the ethical modern day eugenics, um, we conducted a study a few years ago, just a not a genetic study, but just a public opinion survey. We did a national representative sample to ask how people would feel about a fertility clinic offering this service. How would they feel about a school asking for your genetic data or a saliva sample as part of an application process? How would you feel about insurance companies giving you a discount to get your DNA and then risk adjust the premiums based on what they perceive as the risk, which think is all going to happen in the not so distant future. And what shocked me was that I thought that people would be the most freaked out about the IVF clinic because that's like playing God. It's seems so dystopian. Maybe people hadn't seen Gattaca like I had.
It's a good movie. I mean, it's
Yeah.
it's a fun movie when it's sci-fi. I don't want it to be a docuseries, but
Right. So, but what was surprising to me was that we had a benchmark question about whether or not it's okay to judge people on the basis of their genetics. That's a standard question in the General Social Survey and other big opinion surveys, just to have a benchmark. And like those other surveys, about 90% of people said, or 92% of people said it's wrong to judge people on the basis of their genetics. Then they went right ahead and said, sure, let's select babies, our embryos based on their polygenic index or education or whatever,
right.
and they were fine with that. That shocked me. But what, in retrospect, I should have figured was that the when we gave them scenarios like using it for sperm and ova donors, like to have more information about a donor that you might use, or using it for your IVF. They were gung ho about that because that's empowering people to make their own choices, and giving them more information. But when we asked about school using it to make a decision about your kid being admitted to preschool or to first grade or whatever, or college or whatever it is, or an insurance company adjusting their prices based on your genetic risk, they were much less happy. Still, I think a majority were okay with it, which shocked me too. But the divide was: do I kind of am in the driver's seat with this information, or is a faceless, nameless institution judging me based on my DNA? And that's where we saw the divide. And there was no partisan at that time, at least. There was no partisan split on this. There was the only demographic factor that seemed to predict whether people were comfortable with polygenic scores being used in these ways was age. And it's like younger people were more comfortable with a new technology than older people. So yeah, I think we have a window now, because this is unlike vaccines or masking even, or it has not been politicized yet. We have probably a very brief window to have a discussion, kind of a sane discussion about, do we want regulations about this or not? We currently have one law at the national level, at least, the Genetic Information Non-Discrimination Act that Bush signed in 2008, and that prevents the use of genetic information in hiring and employment decisions and in health insurance and healthcare. It doesn't say anything about auto insurance, long-term care insurance, life insurance, school. Yeah,
that's crazy. Sorry, that's crazy that the current law is from Bush 2008. Like, all of the science we've done since, I mean, you mentioned in the beginning, since your book in 2017, and like we're in 2025, and our current genetics changes like every 3 to 6 months, and we have something from 2008. That's a little bit astounding.
And that was written in a time when the thinking about genes was more like the Huntington's or Tay-Sachs era, or sickle cell. Like, oh, okay, someone might have a single gene that makes them different. Not that employers might want to maximize productivity
Right.
by
Right.
measuring a polygenic score. So yeah, if we updated that, and like I said, I don't even clear. There's very clear public policy answers. Like
Mm.
it's complicated. So let's take long-term care insurance. Um, the point of insurance is to pool risk, right? Like
Hmm.
if we all know exactly what we're going to get, then you just save up and manage the risk ourselves. But the idea is that we don't know, and we want to pool information and be covered if we're in the bad end, and what we're willing to pay
Mm.
and lose money if we're on the good end and never need the insurance.
Mm.
Um, at the extent to which the insurance companies can risk adjust, they're kind of and making it more expensive for people who are at higher risk than people who are at lower risk. That's increasing their profits, but it's also uh, kind of hurting the insurance aspect of it. On the other hand, like if we just extended GINA past an amendment, reauthorization with an amendment to say um, life insurance and long-term care insurance can't use genetic information either. Well, that seems like straightforward, but it's not such an easy solution either because today um, millions of Americans have, I think it's up to now 40 or 50 million Americans have access to their own DNA through these direct to consumer genetic testing
Hmm.
services like Ancestry or 23andMe. And that means they can just take one more step, which is, right click and download their raw data, then upload it to a different site and get their polygenic
Hmm.
index
Hmm.
for Alzheimer's or other related dementias. And if they see that they're at the top end of that risk pool, then they would buy insurance. And the people who see that they're at the bottom end would not buy insurance. And granted, it's a noisy predictor at the individual level, but that could create a death spiral in the insurance market because
Mm.
you only get people at high risk buying the insurance, and then the price goes up, and it just becomes a forward feeding cycle. It's been known to happen in terms of certain insurance markets become lemon markets.
Mm.
So it's not clear to me that just banning the on the supply side is the only solution.
Hmm.
Um, Yet these are going to be complicated debates we need to have rationally and not just with knee-jerk kind of ideological responses from both sides.
I'll say on this real quick. I had you may have read it a couple years ago. It came out this book As Gods by Matt Cobb, and he wrote basically a kind of modern history of what we've done in science. He covers eugenics, but it's not just eugenics. It's a lot of things that have been discovered in science on the genetic side of things, and how ethical considerations and then public policy are always lagging behind. It tends to be very reactive. And he goes throughout the 40s and 50s and 60s into the 80s, I think, 90s. And it's a really important history because we can look at that and say, I had him on and we talked about it, we're in the middle of some of that now. And then you pair that also with, the machine learning AI thing as well. I mean, these things have to be proactively thought through. We don't want to get into oh holy shit moment, and then we have to try and fix a crooked line. I mean, it's some really big stakes there. The GWAS studies.
Yeah,
Tell us about this and the importance of them, or not.
um, yeah. So uh, yeah, GWAS, genome-wide association study. Instead of um, uh, focusing on a specific gene, which is how we used to do it, I guess 15 or 20 years ago, where we know this gene in mice, for example. If you switch it on or off, they have these. It improves their cognitive ability in the maze, or it decreases it, or it improves their cardiovascular outcomes, or makes it worse. And we share that gene, so we go in and we test human variation in that gene. That's called a candidate gene approach,
Mm.
and that turned out to be an utter failure in terms of replication, scientific replication, because as we later learned, and I've already mentioned, um, most of the effects in humans tend to be spread across, like teeny effects spread across the entire genome. So focusing on one gene was not really
Hmm.
getting us far. Um, one gene at a time. So the GWAS just throws the spaghetti against the wall and sees what sticks.
Mhm.
It tests for what are called single nucleotide polymorphisms. These um, locations across the genome where I might have like TT, and you might have AA or AT in that location. It just looks for that common variation,
Mhm.
which is about 1%, sorry, 1/10 of 1% across the genome, which is one of the common, I think the reason we say we're all 99.9% related, and looks at those variants and sees how which ones seem to matter more or less. Um, they can almost all go into a polygenic score because um, we, we seem to predict better when we throw all the spaghetti back into the pot. Um, uh, but there has been a critique of this era of genomic science that we
Mhm.
don't actually learn any genetics, meaning we don't learn any pathways or usable information from just this these associations, statistical associations with these little tweaks across our genome. Um, we don't know how they matter or why they matter under what context. And I would say that's partly true and partly false. Um, so, for example, in the educational attainment GWAS, um, we did learn we did learn some things about what drives sort of intellectual success. So there had been one theory that um, myelination, that is the white matter on your neurons, how much white matter or how efficiently your white matter is deployed on your neurons, um, is related to differences in cognitive ability in the population. That because it's the white matter, the myelin, that speeds up the communication, long-term communication in neurons, makes it much faster, the electrical signal. Um,
Which, just for listeners, in something like multiple sclerosis, it starts to the myelin sheaths start to disintegrate. They start to have they become fractured. So the way I learned it was, the fatter they are, the faster the information gets there. So if you're having deterioration because the body's working against itself, it's a complicated process in multiple sclerosis on white matter, you will have limitations in many things, including cognitive abilities.
Yeah. So that one theory is a variation in the thickness, as you mentioned, of myelin was what could explain why some people are sort of more cognitively agile than others
Hmm.
in the population. But in the GWAS, turned out that those genes weren't implicated in that they weren't high up on the sort of weight of the polygenic index. But what were higher was genes that are involved in synaptic plasticity,
Mm.
the formation and pruning of connections between neurons. So, we learn some fundamental biology there through the GWAS approach? But I would say that one of the one of the frustrations is going to be for the biologists that want these sort of you know gene A causes this cascade to gene B and all the way down, and they can trace the whole pathway. Most outcomes are not going to work that way. Um, Partly because, as I mentioned at the outset, um, a lot of the causal pathways work outside the body. They work through the environment. They work through um, anything from sort of the environments we choose to enter or experience to the ones we evoke in response to our behavior or how we look or what have you.
Mm.
Like all of that is genetic effects, but they're happening as they're what we call an extended phenotype
Hmm.
outside our body,
Hmm.
and um, that's just not going to satisfy the sort of wet lab bench biologists who want to be able to do everything in a petri dish, so to speak.
Yeah. I actually want to jump right to the kind of uh, the gene environment interactions. Before we get there, and of course, this could totally derail things, so I don't want to spend too much time on it. But I think it's important. You mentioned in the book, and I think it will actually be a nice bridge into that part of the conversation. So, extended footnote here. Charles Murray and Herrnstein. They wrote The Bell Curve, 94, I think it was. And, you have some criticisms about that data and some of the conclusions made. You're not the first and probably not the last person to have some criticisms about the bell curve. For you, what are some of those criticisms? And more importantly, why is it important to, kind of lay out, respectfully the criticisms on the data, for understanding some of these interactions, especially within group differences, between group differences, things like that. What are some of the ones that you most think are important?
Okay, well, to go back to that book, they made a very sort of kind of seductive argument that in the latter half of the 20th century in the United States, a lot of structural barriers had been torn down. You had the civil rights legislation in the 1960s. You had the decline of kind of the WASPy old boys networks at like elite schools, and that things had become more meritocratic. But the irony, they argued about more meritocracy, was that what determined who ended up on what rung of the socioeconomic ladder in the United States was now more determined by genes than ever before. That's the kind of fundamental argument of the book, I would say. There's a lot of side notes and so forth, but they claim to show that cognitive ability was predicting, and therefore genes was predicting economic success to a greater extent than it had before. So the most easiest sort of critique of that is, number one, they didn't have genes back then when they were writing that. They were writing about test scores taken in high school, which themselves are a reflection of both genes and environment. And in fact, as we talked about in the beginning of the podcast, at that age, the lion's share of what determines our test scores is environment. It's only later in life that genes become the majority of what explains how we do on a test score. So they're not really, they could be picking up inequalities in environment that are driving those test scores, and which in turn drive wages. Second, their trend that the test scores increasingly matter for success was based on 7 years of data from the NLS between, I think it was 57 and 64, 1957 and 1964. So birth cohorts. That's a very narrow 7 years. I mean, you could pick 7 years of Obama and then the 7 years since Obama, and that would be two widely different worlds we're living in. So that was the post-war boom. Like, who knows what's going on at that time? You need a much longer time series to make some sort of definitive statement about these big structural forces and these trends. So, I mean, those are two data critiques that I would say that I hadn't seen as much in the extant critiques of the book. Fast forward to, I think it was 2019 or 2020. It was right before the pandemic started, actually. I remember I was the respondent to Charles Murray for his new book, or newer book. I think he's had one since then, and that's called Human Difference. And there he now has genetic data, because it's 25 years later, and he tries to argue that race differences in outcomes like test scores or in economic success are tied to these single nucleotide polymorphisms, these polygenic scores. But the problem with that argument, there's a data problem there too, is that polygenic scores or polygenic indices are only useful within group. They're only useful within a certain population. They don't explain between group differences at all. And in fact, the polygenic indices that we generally use have been calculated on people of exclusively white European descent. And there's what's called the portability problem. They do not work in other populations. If you take something like height, even the most biophysical outcome, and you use the polygenic indexes trained on white people, and you try to predict height in people of African descent, and it has almost no predictive value, and it predicts the mean height to be, like, I think, 6 inches lower than they actually is in the world. Like the entire distribution of the African descent population is to the left of the entire distribution of the European descent population, which we know is not true. So it's just they don't work. There's actually a lot of scientists working on why is it that these population differences they don't port across population differences. They don't port from whites to Asians, Asians to whites, and so forth either. And part of it might be what's called genetic architecture, but a part of it might be that what I was talking about before, that we're talking about fundamentally different environments that these populations are experiencing. And since the environments are part of the pathway, that the extended phenotype of the genes working beyond our skin, that those pathways are very different, or blocked, or suppressed, or not suppressed, and enhanced in certain populations. And that's why it doesn't predict well. So we really can't draw any conclusions about about these kind of hot button debates like racial gaps in test scores or something like that from polygenic indices.
Yeah, those are nice critiques. I'd say that I think with Charles, I think it's I think my reading of it is that he does tend to lean, I'd say, and maybe overemphasize the genetic component. He has always said, from what I understand, he does not deny that there is environmental influence. But I think most of the critiques of him, which I think are some fair critiques, is that he will downplay it or minimize the amount of environmental impact. So let's talk about kind of really the heart of the book, which I thought was a lot of fun. And this will really, people will talk about this like, oh, well, it's genes and the environment, or it's genes and, social cultural. So your emphasis is, again, is not the inverse where you're just going to go full social constructionist and say like, yeah, genes don't matter, whatever, it's all environment. You don't make that claim at all. But rather you do emphasize, or you do take time to talk about what that looks like with, in association with genes, which is really nice. So you talk about these gene environment correlations, and some people, I certainly remember learning about this in grad school and in physio and even studying for exams about the kind of active niche picking, the passive, the reactive / evocative types. I can even remember trying to memorize the three major ones, and it was always hard to find like a tangible example for each. They get a little bit difficult. But yeah, tell us what these gene environment correlations are. You can go through the three, and why they're really instructive in knowing the impact of environment on genes and what the correlation is there.
Yeah. So active gene environment interaction, I think, is the easiest to grasp, which is just that your genes cause you to either shape your environment or choose an environment that then also has an effect on your outcome. So the people who are good at have the two fast twitch muscle genes, they migrate to the track or the basketball court, and they get the environmental exposure of competition and playing and practice, and that's what I was kind of talking about before, where there's a pathway from your genes to the outcome that requires environmental interaction, and that's active G by E. Or with the kids who don't have those genes, or have genes for spatial ability, migrate to the architecture school or whatever musical talent. You can just play it out. That's we all are selecting ourselves into different environments based on our genes all the time. Even in our most intimate decisions, we seem to be doing that. So, for example, overall, we find in our studies that we're as related to our spouses as first cousins once degree removed, 1 degree removed.
I remember reading this in the book. It was really interesting.
Yeah, which is kind of shocking because we think of ourselves as like very quote unquote modern. We don't we don't marry our cousins anymore. Like in Pakistan and the UAE, there's still about 60% first cousin marriage.
Have you read this research by Joe Henrich? He's a big anthropologist over there at Harvard. He's done a lot of this cultural evolution research, and he's looked at kind of the coordination between first cousin studies and how that pairs with religion and certain history and things like that. And you can see the kind of it is interesting that for a long time in our anthropology, we did have that, and it, as you say, it just still goes on today in other parts of the world. When I read that, that wasn't as surprising to me. As I know we're kind of modern, things like that, or maybe we're postmodern in some ways, but that wasn't as surprising to me as maybe it might be for some people. I was like, if we have that in our history for a long time, there's some code in there that makes it familiar.
Yeah, well, what I find interesting about it is that in the old days, or in not so old days in some of those societies, as I mentioned,
Right.
It was from constraint that we ended up marrying our first cousin
Mm.
because we lived in villages, and you were basically cousins with everybody.
Mm-hmm.
Today we have literally almost a limitless pool of suitors on these online apps and so forth.
Mm-hmm.
But because we have so much choice and we're able to pick
Mm-hmm.
so precisely, we end up doing the same thing. We end up picking somebody who's genetically similar to us. So if you look at, I mentioned, overall our genomes were like cousins first degree removed. But if you look at specific genetic signatures like PGI for education, and we're more like first cousins, full stop. And then if you look at something like height, we're like half siblings marrying each other. So I find that super interesting because that means the next generation there's going to be more genetic inequality when when we're not randomly mating. So that's a form of active gene environment correlation as well. I mean, it's taking us a little side.
So no, it's super interesting though. It's very interesting.
Off the three types, but that's the point of active G by E is that we're not randomly distributed across our environments. We like people with higher educational PGIs tend to migrate out of depressed coal mining towns in northern England and migrate to London. People with higher PGIs tend to move countries across the world even. So we are sorting ourselves based on our genetics, and that's active G by E. But a passive G by E is when your genes, probably because of the constraints or the choices of your ancestors, also expose you to an environment, but you didn't do it yourself. So an extreme case of that in the animal kingdom is the cuckoo bird, which is a parasitic egg layer. Like these chicks wake up and they're not in their actual parents' nest. Their parent sort of, because of their genes, puts them in, disperses their eggs into different nests so that they'll be raised by other birds. In humans, we find that there is the non-random distribution of genes across environments based on who's living where in the generation before. So another example would be that, like, if you have a parent who is prone to depression, you might inherit those genes that are also prone to depression, but you're going to inherit an environment that's prone to depression because you're going to have a listless kind of depressed parent in the household, and that's going to inculcate you for depression itself environmentally, but it's also going to be correlated to your genes because they're going to go hand in hand because of the nature of how we pass on both social environment and our genes to our children.
This is like the kid that is raised in a home of all, or not all, but like athletes, and they're all doing sports or a particular sport, and they're just around the environment all the time, and so there's this passive kind of interaction there where it's like, well, it's familiar, or what is it? What say you study sometimes where it shows, what are the IQ points or academic attainment of, people that grew up with how many books in their house, right? They saw books everywhere, and it was bookshelves, and they went to the library and all these things. That is a kind of you're kind of presenting to a kid, or in development, these environments. As, of course, no one can choose the environment that they're in per se, but these types of things are kind of the passive sense, right?
Yeah, exactly. So what makes it a gene environment correlation is the fact that it's not random with respect to genes, who gets a lot of books in their household and who doesn't. It's correlated with the genes for reading, let's say,
Mm-hmm.
and and yeah, so that these forms of gene environment correlation are make it tricky for us, sort of genetic and behavioral scientists, to separate out what's the effect of genes and what's the effect of environment,
Mm-hmm.
because they're 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 not just they're not independently sorting. So it's hard to nail down what's the effect of one, what's the effect of other.
Mm.
But as I show in my book, in some ways that's the wrong question, because these even these passive environments, much less the active ones, are important parts of the pathway by which the genes have their effect. The last one is that you mentioned is evocative gene environment correlation, and that's when there's certain genes, uh, not by your choice, like in sort of the active case, but in your genes cause a response in others. That's the idea of evoke a response in others, and that colors your environment and affects your outcome. So, an example of that might be the genes for skin tone in the African American community. We found, for example, that even among siblings, one, because of the luck of the genetic draw, is born with darker skin or lighter skin than their other sibling, and that that skin tone in turn generates different levels of stress in those people because of how they're treated, and different rates of hypertension between siblings, of high blood pressure.
But this would be sort of on the phenotypic lines of things, right?
Yeah, I mean, it's the genes for skin tone
Right.
working through the skin to evoke a response in the environment that feeds back on the biology of the body. So that's a evocative gene environment correlation. Uh, you know, like the simplest example I can think of is that beautiful people get more attention or are thought of as smarter, so they're treated differently. They get a lot more attention, even from being an infant. So, the beauty might be caused by the genes, but the social response has an important consequence to it.
Another footnote here. Again, you could talk ad nauseum about this, of course. And again, it feels very hot. It feels very, people like it. I've not been as convinced. I'm kind of waiting. But epigenetics, people talk on and on and on about this, and I feel hesitant about that. Just not because it's not a thing or that it doesn't have potential, but I just don't know if it has the explanatory power that people want it to have. What do you see are some of the differences between GWAS and the epigenetics, and what are some of the they're doing similar things, but they are different. What do you see as the differences there?
Yeah, that's a great question. Epigenetics are the study of the little chemical marks that get attached to our DNA, that are like switches that help turn on or off whether the genes get expressed or not, meaning they get transcribed and then translated into proteins. So they're not determinative, but they definitely influence how genes end up mattering or not. So epigenetics are critical to our biology because you start with this as a single cell, and that's got in every cell that divides from that single cell has exact same genetic blueprint, but yet the cells on your skin are very different than the cells in your liver from the ones in your brain. So it's epigenetics that cause the cells to be different. From from the same basic blueprint. Um, but what has gotten a lot of social and behavioral scientists really excited is the fact that we can trace how the environment, the external environment, can actually affect your epigenetics. And that's an exciting new area of research. For my purposes, up until very recently, it has been less direct focus of my research because I'm really interested in nailing down cause and effect, and genes are really helpful in that regard because we know they don't, by virtue of the fact that they don't change once you're conceived as a zygote. You know, we know that your genome is fixed. So we can. So um, if all of your friends happen to be smokers, and you have this genotype for smoking, um, we know that your friend smoking did not cause your genotype. If there's any correlation, it's got to be going from your genotype is maybe obviously through your own behavior causing you to have friends that are smokers or cause your friends to smoke. So for me, from a perspective of a scientist studying human populations, genes are really useful for sorting out cause and effect in social networks and in families and so forth. Um, Epigenetics are an important pathway like they're an intermediate step between your genes, your and whether you're a smoker or not, how far you go in school, whether you have cardiovascular disease, how tall you are. Epigenetics is an important pathway, and the environment comes into muck with that pathway. It's interesting, but doesn't solve the fundamental problems that I'm interested in terms of really nailing down cause and effect in human life. What I find epigenetics more interesting or useful is as actually an outcome. There are what are called epigenetic aging clocks that can tell you whether you're at a cellular or molecular level, you're aging faster or slower than your years on the planet would suggest. And that's become a really important, exciting sort of quasi outcome for aging research. So now, instead of like if I wanted to give you metformin and give a control group, not give them sugar pills to see if metformin, which is an anti-diabetes drug that's suspected to sort of slow down aging, if it's going to work. I can't wait. I don't have to wait 50 years to see if who croaks first. I can have an epigenetic measure of aging to see in a much more shorter time frame that's realistic for like drug development and so forth, to see whether or not it's having an effect on your aging process. So that's to me like a very exciting, useful aspect of epigenetics. But for what I'm talking about gene environment interaction, it's an important molecular mechanism, but it's not something that I find necessary to get into the weeds of.
One of the last questions I want to ask you is really building off of what we talked about with the gene environments, but is really your kind of main thrust of the book is genes of others within a social environment, the social genome, which is this is the title of the book, affect people. They affect each other. How do the genes of others impact and matter to other people? Sort of. I mean, there's the general like, duh, yeah, of course they do. But like the specific kind of more causal thing, which you're talking about, you can give an example of maybe depression with married couples or with parents, and you'll hear colloquially people will say, "Oh yeah, I got diagnosed with depression. My parent had bipolar, or they were depressed. So it must be the genes." Or oh my mom was an alcoholic, and I got a drinking problem. So that genes, like people will say that colloquially all the time, and the truth of it is to be questioned, I guess. But I guess the larger question, though, is sort of the one you're asking in the book: is how do genes of others within a social environment affect each other?
Yeah, that's I think a really exciting area. What I mentioned before is social genetic effects or genetic nurture is another term for it. Indirect genetic effects is a third term. Um, and there was a really interesting study of mice where Emily Bode and her collaborators, like, can randomly assign mice to different cage mates. So they put mice in pairs, and they had different genotypes they knew about. And for uh, a lot of outcomes, like I think a third of the outcomes, she had 128 phenotypes she measured, and I think for a third of them, they found social genetic effects, like that not just your own genes, but the genes of your cage mate mattered. And for some things like anxiety and depression, the effects of your cage mate were actually bigger than your own genes in your body.
Wow.
You can imagine, like, if you're living in a prison cell, and your roommate, so to speak, is hyper aggressive, that's going to matter much more to your anxiety level than anything about your own genome. If you're a normal person who's worried about your personal safety. And that's what was happening with the mice. So in humans, now we've shown this. Augie Kong, who's a Oxford statistical geneticist who kind of did one of the blockbuster papers on this topic in 2019 in Science, showed genetic nurture of parents, meaning that, yes, the examples you were giving, yeah, I'm an alcoholic, but, blame it on the genes because my parents both drank a lot. Well, it turns out that they gave you the genes for dipsomania, like, through biological inheritance, but the fact that they had genes for alcoholism or addiction, that they didn't pass on, created an environment, that deeply affects your likelihood of becoming alcoholic too. It's an example of what we were talking about before, passive gene by E. Um, but what's really exciting in the polygenic index era is that now we have a tool to measure all these social genetic effects. And as I mentioned before, they're not always like intuitive. So, for example, we found in studying high schools, it wasn't who your best friends were about their genetic influence on your smoking behavior. It was whether or not in the entire grade there was just a few people with extreme values that drove the smoking behavior of the entire social network. So, like, that's telling us something about how social genetics effects work for that outcome, in that population of adolescents. That's very different than how it works in a family. So in a family, we know that there's social genetic effects of parents, like, like, of course your parents' genes through their behavior affect how you turn out. But what's surprising is so far we haven't been able to show that there are effects of your siblings. I mean, most people would think their sibling, especially an older sibling, might be a very formative influence on who you turn out to be. But so far, we're not able to show that. So, yet I also mentioned the example of kids affecting their parents. The kids' genes that are randomly assigned to them from their parents' pool turn back and have influence on their parents' behaviors. So, what I find really exciting about social genetic effects is now we have a tool to study social processes that before we had no. Yeah, we know people in the family were correlated. Everyone was happy or everyone was sad in a given family, but we couldn't trace out the causal pathways. You mentioned the example of the spouses. There's an old adage that says, happy wife, happy life, or the more modern, gender neutral one is happy spouse, happy house. And we went and tested that to see if there was asymmetry in the genes of a spouse affecting the other spouse's mood or likelihood of depression. And what we found was that each spouse affected their genes for depression, affected their spouse's likelihood of depression about a third as much as that spouse's own genes for depression did. So, and it was symmetrical, at least in this day and age. It might have been in 1902 when that first that little ditty was coined, "Happy wife, happy life." It might have been asymmetric back then, that the wife's gene caused the husband mood more than vice versa. But today it's symmetrical. And so, yeah, there's social genetic effects, genetic nurture all over. our social world, but sort of I don't think that's surprising, but I think how it manifests in specific instances is really interesting to learn.
Mm, Yeah, absolutely. Well, I mean, there's more, obviously, much more research to uncover and discover and to work on. I guess my last final question here is where do you feel the future of kind of behavioral genetics or sociogenomics? Where is it going? Whether certain questions that people are asking, and how can we tackle that? It definitely is how can scientists tackle that? Um, Ethically and with good safeguards for when these things start to become more public and more in use.
Yeah, that's a great question. A tough question. I'd say that there's many directions as the research and thinking is going now. So one is to get more and better data. Um, you know, Mostly we've been running our GWASs, as you call them, um, off of what are called SNP chips. That
Hmm.
the chips that measure usually like about a million or a million and a half um, SNP, single nucleotide polymorphism,
Hmm.
but there's really many more variants across the genome. Those are just the ones that are common or tagged in these for efficiency in these chips. And now full genome sequencing has gotten cheap enough just to scan the entire genomes of sample members,
Mm.
and that's going to be exciting to have all the data to sort of improve the predictive accuracy of these scores. Um, Second, and getting more and more people
Hmm.
to study will also improve the scores. Second is that we need within family data, meaning
Mm.
it's much more useful to study, like say, compare siblings or to have um, parents and children in the data set together, because then you can really rule out environmental.
Mm.
You can separate out environment and genes in order to study their independent effects and how they work to come back together. When you just study sort of like random people, like your genome versus my genome, um, we're conflating a lot of what's genetic and our different environmental experiences, as I mentioned, because of gene environment correlation. So, family based data solves that problem a lot. So, moving towards that will be very exciting. A third area is solving the portability problem across populations. That we have a decent amount of data on people of European descent. We're starting to get more data on people of East Asian descent because China and Japan are starting biobanks and investing a lot of money in collecting data. But we don't have a lot of information about people from other parts of the world, Africa, Middle East, South Asia, Native American, which means like Latino
Hmm.
and Latin American populations. So, um, from a scientific perspective, that's just really interesting to get more data on more populations and understand the differences and similarities. And from a pragmatic point of view, if we're creating a utopia or a dystopia of genetic prediction, as I mentioned at the outset, um, currently that's a dystopia or utopia for white people, um, because those are what the scores work for.
Mm.
So if we want to have, for example, or not create a new form of health inequity, let's say in terms of early screening for cardiovascular disease, we would want to have scores that work for other populations as well.
Mm.
So um, those are important areas, I think, in terms of the research. And then in terms of the policy, as I mentioned, we currently have no debates about how these scores should be used, in what contexts are appropriate or unethical. And I think we really need to, as much as I'm, think the AI is a revolution that we need to deal with and think about, so is the social genomics revolution as well.
Mm. Well, Dalton, this has been really, really, really wonderful. I greatly enjoyed this conversation. Your book is great, and you're offering such a wonderful perspective and important research for important and much talked about topic. So keep doing your research, and yeah, thank you so much for sharing your wisdom with us.
Thank you so much, Xavier, for having me.